But okay, so we'll just get started. So as you can tell from the title, I'm going to be talking about nutrition, specifically how it relates to cancer metabolism. And for my title slide, I chose this very lovely random DNA molecule made of a bunch of random vegetables and fruits, because why not? Every nutrition lecture needs that. But anyways, moving on. So to cover the objectives and outline what I want to talk about today, we're going to start by highlighting the link between obesity and cancer. And specifically, we're going to go into detail more with understanding how cancer cells undergo metabolic reprogramming. Is everything okay? Okay, I didn't know if I heard something like that. But anyway, and then we'll move on to further recognize how inflammatory pathways are also involved with the cancer cell progression. And then at the end, we're going to recap and cover like some of the main points, and then also conclude by discussing how this is relevant to public health, because of course, as a public health major, I have to finish with that. Okay, so to start off, I want to bring up the schematic that discusses and shows very clearly the hallmarks of all cancer cells. So this is a pretty old model. It's from a paper from Hanahan and Weinberg from about 2 decades ago, like 20 years ago, so relatively old. But the reason I thought it was important enough to bring up is because it's a very well-established model that kind of shows all of these, especially biochemical hallmarks that all cancer cells will exhibit, and it gives a really good way to understand this visually and to be able to see how these all play out and how these can interact as well. So a lot of these are hallmarks and characteristics that most of us know from studying cancer and understanding it to an extent. So of course, there's inflammation in the body. There's also telomerase involvement leading to limitless replication in cancer cells, metastasis, angiogenesis, etc. So understanding those hallmarks of cancer, I then want to move on to introduce obesity. So we all know obesity as a very large epidemic worldwide, but also especially in the United States. And the reason it is such an issue is that it is a risk factor for many chronic diseases, but also it's largely correlated with very or much worse disease outcomes. And this is especially true for onset of cancer and prognosis of cancer. And so from a nutrition standpoint, obesity is sort of defined as the status of nutrient excess, and also overabundant or positive energy balance. And so looking at obesity from that lens and that perspective, you can see and start to understand how biochemically and metabolically obesity is going to induce a variety of systemic changes. And over time, mostly over the past couple of decades, a lot of research has actually shown, and in the literature it's been shown, that obesity will in fact impact and affect every single one of these 7 hallmarks of cancer, showing again and verifying that link between obesity and how it can reduce or how it can result in worse cancer outcomes. And so I'm not going to be going through all 7 of these hallmarks. As I mentioned in the objectives, I'm mainly going to be focusing on these 2. And first, we're going to start by talking about metabolic reprogramming. So this will touch more into the Kalin paper, which is one of the papers that I uploaded. And so specifically what we mean with metabolic reprogramming in cancer cells. So firstly, we understand how normal cells, and this is mostly like differentiated tissue in the body, so muscle cells, neural cells, epithelial cells, what have you, they will undergo glucose metabolism to generate energy through oxygen-mediated processes, so either aerobic, so cellular respiration, oxidative phosphorylation. As you can see in the schematic here, this is a very condensed schematic. I'm sure we all remember from biochem PTSD, the whole Krebs cycle and everything. So this is obviously much more simplified, but it goes through the mitochondria, and it is with the presence of oxygen, versus in anaerobic conditions or without presence of oxygen, cells will undergo anaerobic glycolysis, usually yielding a lot less ATP, usually like 2 to 4 molecules of ATP per molecule of glucose. So what happens in cancer cells is that cancer cells will actually undergo what's known as the Warburg effect, or Warburg metabolism. And so what happens here is that tumor cells, or cancer cells being very proliferative tissues, they will actually switch and mechanistically switch to promote glycolytic pathways instead of oxidative phosphorylation, even in the presence of sufficient oxygen. So even though they have this oxygen, it's known as aerobic glycolysis, or more specifically, Warburg metabolism. And so you can see in the schematic here, most of that glucose, once it becomes pyruvate, it'll actually go into lactate instead of going into the mitochondria to function for oxidative phosphorylation. Hello. And so, my computer's spazzing out, sorry. Okay, so so now that this has been established as a phenomenon that occurs in cancer cells, the question became, why does this even occur? Why or how is this happening in cancer cells? So obviously there's been a lot of research to find this proposed mechanism, and there is one that has been pretty well established in the literature that the Kalin paper goes into pretty good detail about. And essentially, essentially it includes the role of HIF1 alpha, or hypoxia-inducible factor 1 alpha, as kind of the key player in this mechanism. And so to introduce HIF1 alpha, it is a transcription factor. And this schematic here kind of shows how it functions in the body. So just to introduce that, in normoxia, or normal oxygen conditions, HIF1 alpha will be, in the presence of oxygen, it'll be hydroxylated by an enzyme known as proline hydroxylase domain protein. So once it's hydroxylated, it will then conjugate and be recognized by VHL, or von Hippel-Lindau tumor suppressor protein. Once that conjugation and dimerization occurs, the protein can then be tagged by ubiquitination, and then it's targeted to proteasome degradation. And so it is degraded, and then it's not of use in normal oxygen conditions. So then in hypoxic conditions, which I want to say that hypoxia, it's not necessarily like a starved state or like a crisis state in the body. This can be like local, like pretty normal hypoxic conditions that can happen in the body and due to some random occurrence or whatever. But the regulatory mechanism that happens is when there is an oxygen for HIF alpha to be hydroxylated, it's not going to be tagged and it's not going to be degraded. So instead, it's going to be able to go into the cellular nucleus. It then dimerizes with HIF beta subunit, and then this complex is able to act as a transcription factor directly on the DNA, specifically the hypoxia response element. It will then carry on transcription for the target genes, and then the downstream effect is usually, most notably, it'll be angiogenesis, and then specifically glycolytic metabolism, so just shunting through the glycolytic pathway, which ding, 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 that kind of rings a bell as two aspects of cancer cells. So you can see how this might start to play an effect. So even though this is how HIF1 alpha functions in normal body cells, the problem becomes in cancer cells when in the presence of cancer, these cells will fail to downregulate HIF1 alpha, even in the presence of plentiful oxygen. And so a lot of times, or a lot of the mechanisms that are proposed, there's 3 that I'm going to go into detail here, but essentially what happens is some sort of mutation or inactivation will result in increased HIF production or decreased HIF production, so, or destruction. So I'll go into a little bit more detail about some of these pathways. So first, starting with this schematic, this looks very complicated and like there's a lot happening here, like many biochem pathway and schematics usually are, but not to worry. We're just for now going to focus on what I've outlined in red here, which is the PI3 kinase pathway. So essentially what happens here is in cancer cells, you're going to see upregulation of the PI3 kinase pathway. And because I'm not sure about everyone's different like biochem level or like how much you may like recall or remember from biochem. So just to reiterate and simplify this pathway, with PI3 kinase in cancer cells, it's going to be upregulated. So PI3 kinase is then going to phosphorylate AKT complex, or the messenger, which is phosphorylated and activated. So AKT, once activated, will actually suppress TSC complex, and the TSC complex is tuberous sclerosis complex, which it's a coagulation of different proteins, but essentially TSC complex functions to inhibit mTOR, which for those of you who remember or who might not have heard of it, mTOR is essentially, this is the way it was kind of ingrained to me when I was taking biochem, but mTOR means like growth. mTOR means synthesis and proliferation. So with mTOR being upregulated or activated in any way, you are seeing cell growth, you're seeing synthesis of numerous like nucleotides, amino acid proteins, etc. So with this pathway, PI3 kinase is upregulated, AKT is upregulated. So with AKT being activated, you're suppressing TSC, and therefore you're disinhibiting mTOR. So without the inhibition of mTOR, you're getting net upregulation of mTOR. And with a net upregulation of mTOR, you're going to get upregulation of HIF1 alpha and also cellular growth and proliferation. So that's essentially one of the mechanisms that happens in cancer cells with upregulation and activation of HIF1 alpha. The second one goes back to, I'm introducing this schematic here, where essentially there will be inactivation of VHL. So with the von Hippel-Lindau protein, this is supposed to tag HIF for degradation. So if there's any mutation or for some reason inactivation of the VHL protein, HIF alpha will not be able to be tagged, and therefore it won't be able to be targeted for proteasome degradation. So if HIF isn't being degraded, then you're going to have overexpression and overactivity of HIF-1 alpha. And then the third mechanism, kind of similar to the last one, but basically, so reintroducing, if you remember from the first step here, for degradation, HIF alpha has to be hydroxylated. So in bringing that step a bit closer and like looking into that a little bit more, this is actually a schematic that was in the Kalin paper. If anyone saw it, it was at the very end. But theirs was kind of boring and I didn't like it as much, but I liked what it was trying to say. So I kind of made it myself and made it a little bit nicer and more colorful. But essentially what's happening is you have the PhD protein that is supposed to hydroxylate the HIF-1 alpha in the presence of oxygen. But what's also happening here is with PhD type proteins that actually falls under a subunit or family of dioxygenase proteins and or enzymes. And these dioxygenase enzymes are also involved in a coupled reaction of converting alpha ketoglutarate to succinate, which you might recognize these as intermediates of the Krebs cycle. So what happens here though is, so the coupling reaction occurs and these dioxygenases and PhD are able to hydroxylate the HIF-1 alpha. But what'll happen here with is a normal kind of like negative feedback mechanism where if you have increased amounts of your product, then the enzyme is going to be downregulated. So if there's an accumulation or overcollection of succinate, fumarate, or reactive oxygen species, kind of like products from these intermediates going into the Krebs cycle, PhD is actually going to be inhibited, and then this hydroxylation is not able to occur, HIF can't be tagged, and then the whole kind of similar mechanism that it can't be degraded. So just putting that into words, if inactivation of the redox enzymes like SDH and FH that are supposed to kind of convert succinate and fumarate, if those are inactivated due to some mutation, you're gonna get accumulation of succinate and fumarate. That's gonna inhibit PhD by competing with the alpha ketoglutarate. This is going to prevent hydroxylation of HIF-1 alpha. It's going to escape the VHL recognition, and then you're not going to degrade it, and you're going to have too much of it, which is essentially the whole issue that we're dealing with here. So again, this is a kind of a schematic that just puts those three mechanisms back together. This is again from the Kalin paper, but starting at the top, you're going to have PI3 kinase is overactivated. It's going to phosphorylate AKT, and that's going to inhibit the TSC complex, which I just kind of labeled there, those two proteins, tuberin and hamartin, I don't know how you say it, make up the TSC complex. And since these act to inhibit mTOR, since those are inhibited, you're going to actually have net mTOR activation, and that's going to directly result in more synthesis of HIF-1 alpha. And that's going to lead to this Warburg metabolism that we touched on in the beginning, where cancer cells are going to switch to this purely glycolytic approach. And so now that we've talked about kind of the mechanism, there's still the question of why exactly do cancer cells even want to do this? So as we know, oxidative phosphorylation versus anaerobic glycolysis, or even aerobic glycolysis, there is a much bigger ATP yield with oxidative phosphorylation. So you might be wondering, cancer cells want to proliferate and they want to grow. Why would they make this switch like mechanistically and take advantage of this? So one of the proposed and pretty well researched, and like there's a good amount of evidence proving this theory, is that the remaining carbon skeletons that are not taken through the TCA cycle and are instead shunted down the glycolytic pathway, they are used then for other substrates and other synthetic pathways to create cellular biomass for daughter cell production, which when you think about it sort of makes sense, given that cancer cells, they're not really that worried about their energy yield or how much energy they're producing. They're wanting to replicate and proliferate. So to describe this a bit more visually, I'm going to go back to this really complex biochem image and schematic, but instead we're going to be focusing on the right side, so this part in the square. So in the top left, you can see glucose is coming into the cancer cell through a GLUT transporter, and then it's going through the steps of glycolysis. So, and in each of these intermediates, there is some kind of product or intermediate that's produced in this pathway that the cancer cell actually will shunt and redirect to some sort of synthetic pathway. So to go through it one by one and make sense of it, glucose 6-phosphate, which is produced, can directly be taken to the PPP or pentose phosphate pathway. And as we know, that is a key step for ribulose 5-phosphate synthesis, which is a key precursor for nucleotide synthesis. And obviously, if a cancer cell is replicating and duplicating, it needs to produce a lot of nucleotides. Similarly, when glucose 6-phosphate is shunted through the pentose phosphate pathway, NADPH is produced, and NADPH is then key for regulating the redox reactions, specifically the glutathione reductase reactions. And so I wanted to touch on this, or like take a moment here, because I remember when I first learned about this, I was a little confused. I was thinking, okay, like if you have NADPH actually like stimulating the redox control in the cell, isn't that, you know, good for the body? I was confused as to why this was a bad thing. But if you remember, this is happening in a cancer cell. So with, yes, NADPH is good for the redox control, especially with the glutathione synthase path and reductase pathway. However, if the cancer cell is using that for their own reactive oxygen species stabilization, it actually helps the cancer cell with its own growth. And in fact, this specific pathway will actually stimulate further HIF-1 alpha production, which we learned is not good. So another intermediate will be 3-phosphoglycerate. That is a precursor for serine, which can then be interconverted to glycine, and those can go into amino acid synthesis for further protein synthesis. In this pathway, because this is inhibiting oxidative phosphorylation, PDH, or pyruvate dehydrogenase, is downregulated, so pyruvate will be converted through the Cori cycle, shunted into lactate instead. And this was actually something interesting. I tried looking up into more papers. I didn't exactly find the mechanism to explain it fully, so if anyone knows, or that's something maybe post-hoc I can figure out later, but essentially lactate in kind of the realm of cancer nutrition research, it's considered carcinogenic, especially through these pathways, and when lactate is produced in excess, like here, when through the Cori cycle and the glycolytic pathway, it's being shunted into lactate. But that's especially because lactate is a biomarker for inducing metastasis with certain types of cancer. So I didn't know that actually, and I don't know the mechanism behind that, but that's not good either for cancer progression. And then finally, acetyl-CoA that is produced is able to go into a pathway with NADPH for de novo lipogenesis or lipid synthesis, which especially with mTOR activation in these prolific cell growth pathways, the cancer cells gonna want to produce more of these cellular biomass, but for an obese or cancerous individual, that's not really good for the prognosis or outcome. So especially on the topic of obesity, I did kind of want to, after we've gone through those mechanisms of why this glycolytic pathway is bad and how this is upregulated in cancer cells, I want to tie it back to obesity. So a reminder that obesity will, in most individuals, it's going to manifest as these key things that I've put here. So if it's a green arrow, these are things that are upregulated, or these are in higher amounts, so hyperglycemia, higher insulin levels in the individual, versus the red arrows will be something that's downregulated in obese individuals. So there is less adiponectin, which is another systemic hormone, and there's also insulin resistance. So there's less sensitivity to insulin. And so in obese individuals with the with expanded adipose tissue and increased adipose cells or adipocytes, you're going to see also associated increase in many of these obesogenic hormones. And so with the schematic in B, there's a lot of other pathways like JAK-STAT, MAPK, et cetera, that you don't really need to pay attention to, but mostly I wanted to just include this image to show that in the pink pathway, especially, again, with these increased obesogenic hormones and their associated receptors, you're seeing an increase in these pathways that we already talked about. For example, PI3 kinase, result in glucose uptake, glycolytic pathways, but also protein synthesis, angiogenesis, cell survival, et cetera, which again are hallmarks of what we see in cancer cells. And so now that we've covered that aspect of metabolic reprogramming, I want to draw attention to the very last bullet, where in obesity, we also see manifestation as symptoms of increased inflammatory cytokines. And that nicely goes into the next hallmark that I wanted to discuss, which is in cancer and obesity, in both of these conditions, you're going to see chronic low-grade inflammation. And so specifically talking about obesity and that with that disease status, the inflammation is really associated with white adipose tissue expansion. So I included that schematic again, where you can visually see that with obese individuals, adipocytes and fat tissue are expanding significantly. And so there's a number of associations that are also present during low-grade inflammation in obese individuals. But the one that I want to touch on, and I'll go into more detail about the specifics of these later, but essentially adipocytes begin to function as immune organs. So this is another schematic on the left that kind of shows that, but to walk you through it, and I have another kind of more complicated biochem drawing, but I will walk through that step by step to make sense of these pathways. But essentially adipocytes can actually, as they increase and expand in obese individuals, they can transdifferentiate into macrophages, which we know are immune cells. So when the adipocytes will expand and enlarge too far, they will become a larger, like macrophages, and then they will work to recruit and produce even more inflammatory cytokines and inflammatory molecules. And another point that I saw in the literature is that sometimes with this enlargement in these adipocytes, they can actually pass the point of effective oxygen diffusion. And usually what this means is that within like that area or that site locally, usually it's visceral abdominal fat, it will result in a hypoxic state, which we already talked about being inducive for the HIF1 alpha factor, which we know is not good. Also, these increased inflammatory cytokines will encourage lipolysis, leading to increase of free fatty acid circulation, which also stimulates release of more inflammatory cytokines. And it's all this big positive feedback mechanism, which I will go into detail more in the next slide. But another point I want to touch on is that this whole cascade and this whole pathway will also increase insulin, not insulin sensitivity, I meant insulin resistance. That is a typo. I'm sorry, I'll fix that. It will increase insulin resistance in the individual and also chemo resistance. But okay, so this is the biochem pathway that is, again, it looks like a lot going on here and it's a little complicated, but I included the chunked numbers in like the little boxes in orange. So hopefully I'll be able to walk you through step by step. But essentially, first, what will happen is that adipose tissue, as it expands in an obese individual, it will take on this immune cell role and will recruit macrophages and take on macrophagic roles and recruit more immune cells. And some studies point to that a possible mechanism for this happening is that there's upregulated MCP1, which, correct me if I'm wrong, I believe that's monocyte chemoattractant protein 1, but essentially that's how the adipocytes are able to attract and bring in these immune cells. So then moving to step 2, what happens is with these immune cells, you're then seeing increased release of these immunopathological molecules such as interleukin-1 beta, interleukin-6, and especially TNF alpha, which we know is highly associated with inflammatory states. And then so these will bind to their receptors and go within the cancer cell. And then what we're also seeing along with these inflammatory cytokines is also increased free fatty acids. So these free fatty acids will especially form a complex with the receptor TLR4, which is toll-like receptor 4. And all of these together will increase the pathway and activate NF-kappa B, which I am forgetting what it stands for exactly. I think it's, yeah, no, I'm forgetting what it means. But essentially, that protein and that pathway will act as a transcription factor, and downstream it'll essentially result in the perpetuation of more inflammatory cytokine production, more glycolytic pathways, and more HIF1 alpha production. And so moving on to step 4, also with this, with usually these pathways ongoing in the cancer cell, you're also going to see systemically insulin resistance, and altogether this is all going to lead to tumorigenesis or creation of more tumor cells and proliferation. But you're also going to see lipolysis within, if you go back to the original adipocytes that are enlarging and bringing in all of these inflammatory signals and cytokines. You're going to see lipolysis. So that's a big amount of the fatty acids and of the fat tissue. So you're going to get, again, increased free fatty acid circulation. So again, with all of these cytokines being continuously produced, and then they're signaling for more production of these inflammatory cytokines, more lipolysis, increased free fatty acid is circulating in the blood. It's just all around this very bad positive feedback cycle resulting here. And then in step 5, also just to bring in again those obesogenic hormones, you're seeing with increased adipose tissue, you're going to see increased leptin in obese individuals, also increased leptin resistance. So there's going to be upregulation of this leptin receptor on the cancer cells. And then this is also going to result in upregulated JAK-STAT pathways, PI3K, mTOR pathways that we talked about earlier. And as we know, all of this is going to result in more cell proliferation, cell growth, synthetic pathways, and HIF1 alpha activity. So that was a lot, but essentially you can see that it's just this very continuous bad cycle of all of these inflammatory pathways constantly being brought in and then circulating and then basically like a cascade down to produce more of them. And so discussing that, I want to touch on the fact that I talked about how this can result downstream in this chemo-resistant effect. And so this is kind of a blurry image, sorry, but it was taken from the Laschinger and Hursting paper, essentially where I don't know too much about the mechanisms here. It's more so the schematic is just showing that a lot of the characteristics that we've talked about with especially obese individuals and dealing with cancer, a lot of these pathways that are activated and that are occurring in these individuals will actually lead to chemoresistance. And again, my pharmacological knowledge is not very advanced. I don't know a lot about it. And when I looked through the paper, I wasn't able to find too much of the mechanistic explanation. The most I could get is that there's something that, because of these pathways being disrupted during cancer and in an obese state, there's impaired drug delivery with the chemotherapy. But again, about that, Tim, I don't know if you have anything to add or if you know anything more about this, but I just want to point out that a lot of these do also add into this chemo resistance, which is an issue for people who with cancer who are also obese. But that being said, even though if this is all very kind of like, I guess, negative light onto what's going on in obese individuals with cancer, there is a possible upside into the fact that knowing these pathways and knowing that these pathways targets within them have certain impacts in the response to chemotherapy for individuals with cancer, we can also then take the other way that modifications to some of these pathways can actually affect the cancer mortality rate. So one of the most proven evidence of this is actually using metformin, which we all know is a common diabetes drug. So a lot of it's also diagnosed to a lot of obese individuals too who may be pre-diabetic. But essentially, metformin has is able to activate the AMP-activated protein kinase pathway, and downstream, this pathway is able to then inhibit mTOR, the mTOR signaling pathway, and therefore, as we learned earlier, by inhibiting this, we can then decrease or hopefully decrease tumor proliferation. So I believe there have been some studies that have proven that metformin can actually have this effect for individuals with cancer, especially obese individuals with cancer. Another aspect that we could look at is anti-inflammatory agents that can block some of these inflammatory pathways, especially with the increased cytokine production. The only problem with this is that anti-inflammatory agents, especially NSAIDs, can have very adverse side effects. Haha, at me with my Excedrin that I have to take all the time. But yeah, that is one of the more limiting issues with using anti-inflammatory agents, especially for those with cancer. So that's something that I don't know too much about the current research going on with that, but that is a possible proposed mechanism or pathway that can be looked at for a potential treatment method. And then lastly, of course, as a nutrition major, I was very interested in, okay, knowing these nutritional biochemical pathways, what are some dietary regimens that could be actively taken by patients, either as a preventive or as a treatment effect? And so there isn't too much about this. I will say there's an issue with the realm of nutrition research and especially nutrition clinical research where some nutrition scientists are just not very good at proving their science correctly. I'm going to leave it at that. But there is some evidence out there that a method known as calorie restriction can actually show promising evidence. I believe these were preclinical studies that they can reverse and prevent weight gain in obese individuals, not reversing cancer or like reversing obesity necessarily, but just that weight gain, and that this can have a positive effect to inhibit cancer. But there hasn't been anything really that's shown it can directly reverse the effects of cancer or like reverse any lasting effects for individuals who already have cancer. So it's a bit limited, but there has been some promising evidence. And with calorie restriction, it's not necessarily dieting or just like cutting your plate in half, even though that image looks like that. It's like a tiny little appetizer. It's more so that looking at what the patient's normal calorie intake is and being able to adjust it while also adjusting proper macromolecule content so that they're still eating enough to meet their energetic needs, but it's a restricted amount. It's kind of portion sizing, but a little bit more scientific and a little bit more clinical. And so to finally kind of tie everything together, this is an image that Dr. Hursting used during one of our lectures, which I thought was a really good way to just summarize it all up. But essentially, when you have an obese individual, especially with cancer, they are going to have manifestation of symptoms similar to metabolic syndrome. So you're going to see increased insulin, insulin-like growth factor, an increased leptin to adiponectin ratio, et cetera. And so these are going to, and as a result of this like chronic positive energy balance and nutrient intake, you're going to see an increase in the growth factor signaling. So these PI3 kinase, mTOR signaling, and inflammation and inflammatory cytokines. And all of these contribute, as we saw mechanistically, to the increased risk of cancer and worse outcomes with disease progression. But then similarly, again, bringing up the idea of calorie restriction. So with calorie restriction, there has been shown immediate decreases in some of these obesogenic hormone levels, especially in leptin versus the adiponectin ratios. And so with these decreased levels, we can hope to see that mechanistically we will show lower growth factor signaling with the mTOR pathways, lower inflammatory cytokine production, and the hope is that if downstream, it will lead to lower cancer risk and less worse progression of the cancer and of the disease. And lastly, to conclude it and kind of tie it all up in a bow to reemphasize the importance and relevance to public health. Again, we know that obesity is a comorbid risk factor and it's associated with many chronic diseases, but especially with worse cancer progression. So, and this increased risk due to obesity can be attributed to how it affects directly some of the targets and biochemical metabolic characteristics within these metabolic pathways. So understanding this link between obesity, cancer, and inflammation, it can help us to identify more of these additional risk factors that are causing these dysregulations at the biochemical level, but also increase potential treatment options. So for more of an action plan, knowing these kind of associations and knowing these pathways, we need to further research needs to further identify downstream targets within these biochemical pathways that can be pharmacologically adapted for better treatment options or better medications, and also identifying other measures, possibly dietary measures for individuals that are already affected with obesity and cancer, as there are plenty of preventive measures out there, but more so something for someone who already has these issues, and then moving forward to lessen the severity of their cancer prognosis. And that's it.